Hi, as part of Unit 2 Emerging Infectious Diseases and Antimicrobial Resistance module, today I'll be discussing the basics of emerging and re-emerging diseases and zoonotic diseases, models and mechanisms of pathogen crossover across species boundaries, emerging infectious diseases and their relevance in the 21st century. To start with, the 21st century has witnessed several infectious disease outbreaks, which include severe acute respiratory syndrome of 2003, H1N1 pandemic of 2009, Middle East respiratory syndrome of 2012, Ebola virus disease epidemic, which occurred between 2013 to 2016 in West Africa, Zika virus disease epidemic of 2015, and the most recent COVID-19 pandemic, which still continues to haunt us. The understanding what we have gained from these experiences is that these diseases have the ability to cause substantial morbidity, mortality, and socioeconomic impact. And they also have the ability to spread across borders to infect people and communities in multiple countries within a short span of time. Here I will be discussing the definitions of emerging and re-emerging diseases and zoonotic diseases. When it comes to the definition of emerging infectious disease, it is defined as an outbreak of previously unknown disease, for example, COVID-19, a known disease that are rapidly increasing in incidence and geographic range in the last 2 decades or 20 years, Zika or Lyme's disease, or persistence of infectious diseases that cannot be controlled. Dengue is the best example where we do not have a vaccine, and this disease is causing public health threat, as well as it has the ability to cause a very high morbidity and mortality in the community. Moving on to re-emerging infectious diseases, these are the diseases that reappear after they have been on a significant decline. So the best example is malaria, which kind of showed a decline in 1970s due to public health interventions, and then showed a rise and still continues to be a public health threat. And the other diseases include influenza, tuberculosis, cholera, and pertussis. Zoonotic diseases, these are the diseases which transfer from an infected animal to a human through direct contact or through food, water, or the environment. Nearly 70% of the zoonotic diseases are emerging infectious diseases. So both emerging and re-emerging infectious diseases could be of zoonotic origin. Here I will be discussing the four models of zoonotic crossover or spillover. These are the accepted modes of disease transmission. One is direct, where a virus is transmitted from a domestic dog to the human. The best example is rabies. This is through the bite of an infected rabid dog. Next is the intermediate. So the intermediate host could be a vertebrate or an invertebrate. So when it comes to vertebrate, say a protozoa is transmitted from a wild species to a domestic dog, which then can transmit the parasite to humans. The best example is babesiosis. Intermediate by invertebrate. So here the arbovirus can be transmitted from a wild primate to a mosquito, and then the mosquito can subsequently pass on the virus to a human host. Next is the environment, where say a cow is infected with a particular enterovirus. This can be transmitted to humans by contaminated water when cow sheds the bacteria or the virus in the dung, and this could cause infection. Next is the environment. For example, cow species could release the enterovirus such as E. coli into the environment, which could contaminate the water source and which can subsequently infect humans on consumption of the contaminated water. There are three critical questions which we have to keep in mind when we are discussing about the mechanism of crossover. One is what determines when and where a disease will first cross from animals to humans. Which factors facilitate emergence after a successful host shift? How do we apply this knowledge for the prevention and control of diseases? Here I would spend a few minutes in discussing the mechanism of pathogen crossover across the species. As you all see on the figure on the right side, pathway of crossover is a very complex phenomena. So here the first stage is distribution and intensity of infection in the reservoir host. So this depends upon the reservoir host distribution, reservoir host density, prevalence of infection in the reservoir host, and intensity of infection. So the simple part to understand is the reservoir host distribution, basically how these population or the reservoir host populations are distributed in a particular geographic area, or reservoir host density, which basically means the population size. Are these animals thickly populated or sparsely populated is one of those factors. And prevalence of infection. So if the infection is highly prevalent in a particular reservoir host, chances of spillover is also high. And then intensity of infection. Again, the prevalence is dependent on intensity of infection. And intensity of infection is the ability of the pathogen to infect multiple animals in a quick session or within a short duration of time. So this kind of helps in building the prevalence as well as incidence of disease in a particular reservoir host. So the next stage is pathogen release from the reservoir host. So this could occur through excretion, slaughter, or a vector. Following this stage, it again becomes a very crucial stage where pathogen survival, development, and dissemination. So this is a stage where we discuss about the pathogen pressure. So pathogen pressure basically means the amount of pathogen that is available to humans at a given point in time and space. So basically the amount of pathogen at a particular time in a particular geographic area. So following this, again, humans have to come in contact with the pathogen. So human exposure to pathogen has to occur. So this is again dependent on the human behavior mostly. One is humans coming in contact with contaminated source of water. Second thing is butchering, preparing, and eating the carcass meat. Again, this is also a behavior. Next, the biting rates of the vectors. So humans have to come in contact with the vectors to pick up the disease. So say we have a good dose of bacteria and the pathogen has found a route of entry. There is still some structural barriers and our own immunity and molecular capabilities, and then replication and dissemination cycles have to be completed before the pathogen actually establishes in the human body. So the take-home message from this slide is that the mechanism of pathogen crossover is a complex phenomena, and it needs at least pathogen to pass through multiple stages before it actually reaches the humans. And even within the human body, there are mechanisms which can prevent the disease from occurring. Moving on with the discussion on mechanism of pathogen crossover, the pathogen, as I was discussing earlier, must overcome a series of barriers to transmit from one species to the another. So spillover of some pathogens require all the gaps. So these are depicted as holes in the various stages in this figure on the right side. So all of the barriers align within the narrow window in space and time. So basically, pathogen to have a successful spillover, it has to pass through all the barriers. And then if any of these barriers are impenetrable, the spillover cannot occur. So this also gives us a hint that breaking one or two of these part of the transmission chain could help in preventing the disease. For other pathogens, protected survival in the environment will play a major role. For example, the Bacillus anthracis spores. So they can survive up to 6 to 7 decades and cause infection. So again, here the environment plays a role, and the way the pathogen can adapt to that. And then the other one is the widespread dissemination of Coxella burnetii. So this basically depends on the spread of aerosols. So again, here the regular infection transmission might not apply for these spillovers. So here I'll be giving some examples of spillovers and the bottlenecks, and how much understanding we have in terms of these zoonotic diseases, spillovers from animals to humans. So different barriers permit or constrain the flow of pathogens from one species to another. So that is kind of shown in the form of the funnels, and then how these barriers are penetrated. So The width of the gaps in the barriers represent the ease with which a pathogen can flow through the barriers and will vary depending on the context. And the question marks represent the points at which the barriers are especially poorly understood or there are high gaps in the knowledge. So, for example, if we take rabies, so the reservoir distribution and reservoir density of the animal population. So basically, the dogs are quite abundant in the urban areas of India. So whereas the prevalence of the disease or infection intensity or pathogen release. So when we look at these other factors, so they are very constrained. By the time they actually come and reach the humans, there are a lot of constraints the pathogen has to pass through. And then once it reaches the human, the infection kind of establishes, and there is no way the person recovers. So it's most of the time it results in death. So in case of leptospirosis, we see a larger funnel. So the gaps in the reservoir distribution, reservoir density, prevalence, and even the intensity of infection. So when we look at these gaps, they are quite broad, which basically means that the pathogen can pass through these barriers very easily. After that, the pathogen release and then the pathogen survival and spread. So here the constraint starts, and by the time it actually percolates to humans, it's very restricted. So that is one of the reasons why leptospirosis is not highly prevalent in all the populations, other than the incidences where humans come in contact with the contaminated environment with the leptospira. Again, the E. coli, I think this example we have already seen. So the constraints are in terms of prevalence, intensity of infection, pathogen release, and once the pathogen kind of crosses this, the barriers are quite wide. We can actually see that the pathogen can easily pass through, though the understanding is still lacking. So similarly for Toxoplasma gondii, we can see at different levels, we see constraints and ease with which the pathogen passes. Ebola is one of those diseases. We know that the virus is prevalent in bats. So here the first few barriers such as reservoir distribution, reservoir density, to some extent are broader. So after that, the knowledge about how the disease actually gains an entry into the human population is very restricted. And once it reaches the human population, again, the human body as such does not have much of a role in protecting itself. So that is one of the reasons why it's one of those dreaded disease which humans are facing. So this kind of helps us understand the role of various stages through which a pathogen has to pass through before it can actually establish as a human pathogen. So once, say, for example, Ebola virus reaches the humans, so it can transmit to other humans quite easily. In case of a barrier where we have the host barrier, for rabies, the disease kind of is restricted to the person who is infected, so it doesn't pass on from there on, but it causes mortality in the humans. So moving on with the discussion on emerging infectious disease transmission and its relevance in the 21st century. So to give some background, during the pre-modern times, it was the colonization, slavery and war which led to global spread of infectious diseases, and these diseases used to cause substantial morbidity and mortality. So the diseases include TB, polio, smallpox, diphtheria, before the advent of vaccines. In the past 2 to 3 decades, there have been a lot of medical advances and improved access to healthcare and improved sanitation. So this has reduced overall mortality and morbidity due to low respiratory tract infections and diarrheal diseases. The new era of infectious diseases, these are defined by outbreaks and quick spread of emerging and re-emerging diseases. So this is mainly aided by global connectivity, urbanization, and climate change. We look at the way the connectivity has been in the last century or so. What is very striking is that during the post-Columbus era, the connectivity was mainly between parts of Europe and Caribbean. This was the very simple route. And then the other one is the classic, the trade routes. So this was all the wars, so which was mainly confined to Europe, parts of Middle East, and North Africa. So then came the trade, the slave trade routes. So this, which is depicted in red. So we can see that parts of North America, South America are connected with the South Africa and other African regions. So if we look at the 2004 SARS epidemic, so the connectivity is very high. We can see that most of the parts of the world are connected well. So when compared to 1933, where we had hardly any connection. So what we see here in 2020 is that global international air travel network has expanded substantially between 1933 to 2020. Moving on with the discussion on changes to the global disease burden in 21st century. So this is mainly impacted by urbanization, climate change, and demographic changes. So the impacts of urbanization on the infectious diseases is nicely depicted in this figure. So I'll be just focusing on those aspects which facilitate emergence of infectious diseases. One is encroachment of the forest by human population. So with rapid expansion of urban areas, this is leading to more interaction with the wildlife and the spillover risk. The second thing is increasing population leading to poor quality of life. One is with the rise in population growth, more persistent outbreaks occur, and especially the respiratory diseases in urban areas. Second thing is poor sanitary conditions due to high population density. This increases the spread of enteric infections. The aspect which is more scary is that the vectors are nicely adapting to the urban setting, especially the vectors such as Aedes aegypti and Aedes albopictus, which transmit dengue. So these are well adapted to the urban areas and cause huge outbreaks in humans. The fourth aspect, which is also of concern, is the global transit network. So this is increasing the risk of importing and exporting the diseases very quickly from various parts of the world. So moving on to the effect of climate change. So here I'm just briefly touching upon the effect of climate change and demographic changes. One is with the climate change, there is increase in geographic range and survival of vectors. So some of the vectors which were not found in parts of Himalayas, for example, the Aedes species. Now, because of rapid change in climate, we do find outbreaks of dengue in parts of Himalayas also. The pathogen evolution and increased susceptibility is another issue. So with more interactions with the wildlife coming up, and then the changing climate. So there is a increased susceptibility also in the human population to these diseases which are evolving newly. So now the question is, how are we going to tackle these challenges? One is forward-looking research. So basically, we do have institutional structures built to address old enemies. These can be adopted for emerging infectious disease threats. The second thing is increased attention needs to be paid to pathogens currently circulating both in wildlife as well as domestic animals. So this is also a most important part. So basically, we need to strengthen the surveillance in the wildlife as well as domestic animals. The second thing is reinterpretation of information available from classic tools such as PCR, and leveraging the multiplex serological approaches to detect pathogen emergence. New technology, including advances in data collection and surveillance, needs to be harnessed. Increased interest in integrating multiple serological platforms, from genomics to case data, to a better understanding of pathogen spread. In summary, crossover or spillover could occur by multiple modes ranging from direct to environmental exposure. Spillover is a complex phenomenon which is promoted by successive processes that enable animal pathogen to establish infection in humans. Different barriers permit or constrain the flow of pathogens from one species to another, and better understanding of these barriers could help in developing public health interventions to prevent and control the disease. Global disease burden in 21st century is multidimensional. This is a result of demographic changes, urbanization, climate change, and international travel and development of technology. Forward looking research and new technology and interpreting multiple surveillance platforms could be a solution. Thank you.